I would like to welcome everyone again to the webinar today, the perspectives on clinical pharmacology and pharmacometric aspects of development of CAR-T and TCR-T cell therapies. I would like to say it gives me great pleasure in introducing the three speakers today. The first speaker today is Vijay Upreti, with whom I have had the good fortune of working over several years and publishing papers with him. Vijay received his training as a pharmacist from India, and then he earned his PhD from the University in Maryland in pharmacokinetics and pharmacodynamics and biopharmaceutics. He has over 17 years of industry experience. He has worked on small molecules, biotherapeutics, etc. He has worked across TAs. He has worked in oncology, inflammation, cardiovascular, metabolic diseases. He has worked in, Bristol Myers Squibb, Amgen, Dr. Reddy's lab. He has overseen the global filings of several molecules, both small molecules and large, and cell therapeutics, which are on the market now. He currently is at Amgen and serves as a clinical pharmacology oncology therapeutics area lead, and oversees several senior scientists who support clinical pharmacology modeling and simulation aspects of different projects. He also leads a group of experts. They report directly into him, and they oversee PBPK modeling and simulation. He has authored several papers. He has served on several committees. He serves on the editorial board of several journals, scientific journals. So we are very happy to have him speak about CAR-T cell therapies here today. Next slide, Vijay. Our next speaker is Hardik Modi. Hardik did his PhD, master's and PhD from the College of Pharmacy at Ohio State. He has an interesting background. He started out as a Buckeye in the US, but then transitioned and did his postdoctoral fellowship at University of Florida as a Gator. If you all recall Urban Meyer, who started out as a Gator, won his national championship there, and then went to Buckeyes and won another championship. But Hardik did the good thing. Hardik is currently a senior scientist, PKPD in preclinical sciences and translational safety in Janssen. In his current role, he supports early discovery, preclinical, early clinical development of therapeutic biologics in oncology and other therapeutic areas, serves as the PKPD subject matter expert for cell and gene therapies. He has published several articles, scientific articles, in peer-reviewed journals. If you go on to the next slide, Vijay. And I would like to welcome the speaker of the last talk, Dale Miles. Dale received his BSc from Brigham Young University, and then his master's and PhD from the Arizona State University. I think the Sun Devils are the football team there, if I remember correctly. And that's only because, we at Iowa played them a few times. Of course, across his career, Dale has supported multiple approved therapies. He has overseen multiple therapeutic areas. He has worked on small molecules, biotherapeutics, antibody-drug conjugates, etc. He has worked at Exelixis. He has worked at other companies, Pharmacyclics, and currently he's at Genentech. He's been at Genentech for the last 7 years. He's a director and senior principal scientist in the clinical pharmacology department, and oversees several modalities. He has led cross-functional teams, novel modalities, and faster filing initiatives within Genentech. And between the three of them, our speakers have over 100 publications. They have a lot of experience under their belt and a lot of insight to provide us on the talks that they're about to give us. So with that, I would like to welcome them and would request all of you to welcome them. Vijay, why don't you take off?
Thank you, Nag, for an excellent introduction to all of us. And first of all, I want to thank you for being here today. Today, as Nag talked about, are you able to see my screen? I want to make sure that you're able to see my screen today.
Yes, Vijay, at least we can see it. It's in the presentation mode. So.
And I want to make sure that there's some technical error happened on my side. Are you able to see now the webinar outline slide?
Yep, we are on the webinar outline slide, but not the presentation mode.
Okay, I'll do that. Thank you so much. Sorry for these technical issues.
Webinar outline slide, presentation mode.
Thank you, Nag. And again, I want to start by saying thank you to Nag for an excellent introduction. I want to say thank you for all of you for being here today. Really, as Nag mentioned, what we have done in this IQ forum for you is to distill the best of the knowledge we have in the field of clinical pharmacology strategies for optimal development of CAR T cellular therapies. That's what I will get started with. And then I have requested Hardik to talk about PKPD modeling approaches for cell therapies and translational consideration. We are going to end our talk with TCR-Ts, which is a very exciting space. You will hear about from Dale, and then we'll have question and answers at the end. And before I go into it, I really want to say thank you to the IQ Consortium, Clinical Pharmacology Leadership Group, as well as the Translational Sciences and ADME Leadership Group. This really is the IQ forum where both of these groups have come together to put together for you what you're seeing here today. And this is a working group to which I'm thankful to all these members who have been contributing over these years to the knowledge which we are sharing with you today. That's the PKPD team. So a big thank you to all of them. And really, this is the reason why we all are here today, because CAR Ts have brought a paradigm shift in the treatment and management of liquid tumors, and they've shown a great potential in solid tumor space. And what I mean by that is, if you look at the efficacy in liquid tumor space for CAR Ts, it is nothing less than impressive. I'm showing you some of the data for liquid tumors for the first approved CAR T, Kymriah, and one of the recent approved Abecma for multiple myeloma. So two different type of disease indication. These both are relapse refractory. What I mean by that is there's no other option left for these oncology patients. If they're relapsed and refractory, all options have been exhausted. And then look at these impressive response rates, which are over 80%. So almost benefiting everyone, those who earlier were not benefiting with anything available. And not only that, the responses are really meaningful. What I mean by that meaningful is in patients who achieve CR, we also are able to completely abolish the disease to minimum residual disease. That's complete abolishment of disease, and that's a new definition of cure. So these are offering a paradigm shift in the treatment of oncology indications, and these are single dose curative paradigm. The first dosing is supposed to take them over and not require redosing, although that may not always happen, and we'll see those things, but it has brought a lot of excitement in this field. And in solid tumor, there's a lot of space to improve, but we are seeing some encouraging results. Based on these type of results, there are 6 CAR Ts now already approved, 2 in BCMA. Even after Abecma, there's one more approval that happened very recently. So there are 2 anti-BCMA for multiple myeloma and 4 which are for anti-CD19, which is originally how the approval started. And these are in the leukemia space, ALL and lymphoma space. So overall in liquid tumor space, we have a paradigm shift in the treatment, and they're bringing single dose curative paradigm. In solid tumor, we have a promise, but what we are seeing is that there's a great potential to improve them. In solid tumor, one of the problem is not being able to assess the surface antigen, which is typically how the CAR Ts target the mechanism. So we are looking at a different mode where we are targeting endogenous targets, and that is possible through MHC recognition, body's original mechanism of antigen recognition, and that is where the field is shifting to TCR-Ts. And TCR-Ts carry a very strong advantage in solid tumor, where now you can target a lot of endogenous antigens, and that's where Dale is going to talk at the end about the TCR-T space, which is now overcoming about the solid tumors limitations we have seen with CAR-Ts. So overall, a lot of excitement which is developing in CAR T, both for liquid tumors and solid tumors. Now, I want to give you a little bit overview that this IQ forum, the combined CPLG and TLG group, has been working very hard to make interactions with agencies. And we have talked both with FDA as well as PMDA. And this is just a summary of the scientific exchanges we had with this since 2019. And now, actually, we are very excited in this group because in just last month, we have a FDA draft guidance, which is really giving some guidance on development aspects for CAR Ts and cellular therapies. So a lot of excitement and joining of hands both from industry as well as regulatory agencies to bring this really paradigm shift changing therapies to patients who really need them. So we're really excited with how much is happening in this field. So we are really thankful that both industry and regulatory agencies are joining hands to bring this in an accelerated manner to patients. So this is what really I planned in my talk to talk to you about. We'll first briefly talk about the key design aspects, engineering aspects of CAR cell T therapies to give you some background. Then we want to look at the unique PK/PD aspects of cell therapies and the dose exposure response for both safety and efficacy. That's quite unique, and you will see why that is. And then there are multiple factors, multiple factors, and I think you will see that throughout our presentation with all of us talking about all those factors which affect the dose exposure response, starting from the design of the CAR T, as I will start with, to the manufacturing factors starting from the starting material itself being variable to the patient factors, which can affect the dose exposure response and opportunities to optimize them. We're going to talk about that. And then I want to end with talking to you about some of the considerations for optimal designing of these clinical studies and dose selection. So let's get started about that. CAR Ts are gene therapy. They are a gene therapy, but they are different because they're ex vivo gene transfer. So once we take a patient cell, we are going to modify them exogenously, not in the patient. So that's the ex vivo gene transfer, so that we are fortifying these to recognize the tumor specific antigen. And then they exert their mechanism through TDCC, the T cell dependent cellular cytotoxicity. Now that is a common knowledge, but here is what the unique aspect about it. Compared to any other biologic which we know, CAR Ts are a living biologic. So up to now, all the biologics which we know are not living, but they're living, they're dynamic, and during the manufacturing, they expand, and when we dose them, then they undergo expansion. And that's really the reason why they give you single dose curative paradigm, because they're expanding within a patient. Now that's amazing, because it can give you a single dose curative paradigm, but that also brings a lot of variabilities starting from the manufacturing material itself, which is the patient's T cell, which are harvested, then are lentivirus transfected to get this receptor in the exogenous manner, and then are infused back. When you are doing all this, you are introducing factors which are highly variable and impact dose exposure response. And then once they are infused, then the patient factors affect them. So numerous intrinsic, extrinsic factors will affect their dose exposure response for safety and efficacy. So that is one of the challenge. The other challenge which the field has faced up to now, all the current CAR T therapies, all 6 of them approved till now, are autologous. They're derived from same patient. When they derive from the patient, there's up to 1 to 2 months or more delay when you do all this process of leukapheresis, lentivirus transfection, expanding them, quality control, and then bringing it back. There's a long vein to vein time. And that long vein to vein time is something which patients don't have. Time is of essence. So that's a limitation currently, which autologous and all current approved CAR Ts are. The field is slowly shifting to the newer generation, allogenic. Allogenic CAR cellular therapies are coming from healthy donors. And this actually has a huge potential of making CAR Ts off the shelf, just like other bispecific T cell engagers or antibodies where you have off the shelf product handy. That is a great advantage, and it can drastically cut waiting time for patients who don't have that. So that's a very big advantage. But obviously now you can expect, when you're going to take a product from healthy donor and introduce into another patient, you're going to have graft versus host disease. And that's one of the reasons that there's a challenge to actively manage it. But there are strategies being developed, and a lot of work is progressing in this field in allogenic CAR Ts. Also, allogenic CAR Ts make it much easier for redosings, which sometimes patients need because there's a failure to launch of the cell therapy, as you will see in my subsequent slides. It's much more easier to do redosings in allogenic CAR T therapy because it is available as an off-the-shelf product. But again, they are yet unproven because there's no FDA approval yet on allogenic CAR Ts. But the field is very rapidly evolving to get that. And I think that's where we need to all go to bring it earlier to the patients. Now, let's just briefly look at what exactly are CAR Ts, right? CAR T is really a synthetic receptor, which is recognizing the surface antigen, which is specific on tumor cells. Now, in this receptor is linked with intracellular signaling domains to co-stimulatory molecules like 4-1BB CD28, and it has CD3 zeta component. CD3 is really for recognition to the T cell, which is this universal receptor. So it's for T cell activation. But here is the thing. The 4-1BB and CD28, these co-stimulatory domains, have a very big aspect of enhancing the expansion and persistence. So the first generation of CAR Ts which were developed, they lacked these co-stimulatory domains, and they will not have expansion and persistence. They will lack the exposure. Now the newer generation, the second generation ones, have co-stimulatory domains, and these are allowing to maintain the exposure. All the FDA approved CAR-Ts are currently the second generation one, where we have co-stimulatory domains. And the newer generation, which are under development, clinical development, are actually introducing cytokine transgenes like IL-12 on top of these CD28 or 41BB co-stimulatory domains to really enhance with stimulatory cytokines the responses and maintenance of them. So there is an opportunity to improve the dose exposure response through engineering of CAR-Ts also, which is space which we need to work on. Now, as you can all understand very well, CAR-Ts are very different than regular small molecules and large molecules. It's a dynamic living biologic, and the PK is characterized as cellular kinetics of the cells. The pharmacokinetics is cellular kinetics, and it is characterized through using qPCR for measuring the transgene or through flow cytometry, which measures the cell itself. Now, both the methods can be used. Typically, qPCR is used, and when you are using both the methods, you want to show them they are concordant, but they may not always be concordant. And data from qPCR versus flow cytometry from one program to another can be very, very different. And some of the reasons that can happen, just want to highlight it here because some of the times you will see that qPCR will detect multiple transgenes when they are inserted, but flow will only measure one cell. So even though you have multiple transgenes, you may not see that. So flow one reading will be highly underestimated versus the qPCR. And if there is intracellular trapping and downregulation of the expression, then you are not going to have cell counted in flow cytometry, but you will see that in the qPCR. So this may be disconcordance, and something we all need to be aware when we are looking at clinical pharmacology aspects, looking at the methodologies which are used to characterize pharmacokinetics of CAR-Ts. And also, when just done by qPCR, the units which are reported sometimes are not consistent, sometimes being reported as copies per microliter, sometimes copies per microgram. So we need to be, it's one of the challenges where we have to be aware about how to combine the data across CAR-T programs. Not going to spend too much time on this. I want to spend more time on how does the exposure and pharmacokinetics looks like for CAR-Ts. CAR-T pharmacokinetics is multiphasic. And what I mean by that is once you infuse the CAR-T, they undergo a rapid redistribution. They undergo distribution and taken up by the tissues and get completely disappearing from the circulation. That phase is very rapid. But the biggest part is the beauty about what I mentioned to you earlier, which gives it single dosing curative paradigm, is they start expanding. Typically by around 10 to 12 days, you will see an expansion, a big expansion. And if you have not designed the PK sampling very carefully for the earlier part, you may actually miss it out. But the key part is to capture the maximal expansion. And then expansion parameters are typical parameters of peak exposure, the time to the expansion maximum, and its AUC, which are very general, which we all in ClinPharm world and very well understand these parameters. And then persistence parameters are the one like elimination half-life, the last measurable concentration, and the time to last measurable concentrations. And those pharmacokinetic parameters show us expansion and persistence. Now, if you look at the relationship between dose and exposure, which is one of the key aspects of characterization of clinical pharmacology for cellular therapies, general trend across the programs which we have seen is there's a overlapping exposures for doses, and then there's a lack of consistent trend in exposure. And this, if I want to just highlight you for the very first approved CAR-T Kymriah, over a greater than 10-fold range, the exposures were completely overlapping. They had done a dose-based, body weight-based dosing, and there was complete overlap. When they look both the total dose as well as body weight adjusted, there was no relationship for dose and exposure. There was a complete overlap on this, and responses were observed across the dose ranges, and they have approval based on body weight-based dosing. Body weight was not found to be a factor for determining the exposure. And this overlapping exposure trend was, when we look at one of the BCMA, Abecma dose exposure relationship, we were able to see some separation between the doses, but only was seen for the lowest dose. In the doses where really clinical responses were seen meaningful and where the approval is in the doses, there is overlap in the exposure. And actually the trends are not consistent when you look across the CAR-T space, but you will see a general overlap in exposure. That's what I want you to keep in mind, and we'll look at some of the factors why that will happen. But if you look at the relationship between exposure and response, actually you do see a beautiful, predictable relationship between exposure and response. you see that in responders, there is higher expansion, which is characterized by higher peak expansion, Cmax. You see they achieve expansion earlier, typically around 10 to 12 days, versus non-responders in which there is a failure to launch, and then the Tmax really is much more delayed. And then you also see longer persistence in the responders versus non-responders. So these parameters are also consistent across all the CAR-Ts. You see a relationship between exposure response, but you do not see a relationship between dose and exposure. And if you look at the relationship for exposure, it is also correlated with the depth of the response. I talked to you about complete abolishment of the tumor burden, which is minimum residual disease negativity, and that also has been shown to be related to the peak exposure which is achieved. So we are seeing beautiful relationship for exposure response for efficacy. Before talking about the safety, right, I want to bring you this key concept about its pharmacodynamics, that based on the expansion, the helper cells, the cytokines are released, and the cytokine release is obviously the key part of its mechanism of action. It's a signature pharmacodynamic response, is also shown to be associated with efficacy, but is also shown to be associated with safety issue. And this is one of the biggest safety concern for CAR-Ts, is cytokine release syndrome, which is much higher release of cytokines, and this remains to be one of the primary issues which we have to mitigate, manage in case of cellular therapies. And now we have in our management strategies Actemera, which is IL-6. As expected, it's a cytokine antagonist for IL-6. It's approved now in the label for the treatment and management of CAR-Ts, CRS mitigation strategies. So this is something which we all need to keep in mind for CAR-T therapies. There's beautiful exposure response, there's beautiful depth of responses, but also there are safety concerns. The primary safety concern is CRS, which can be mitigated with appropriate strategies, one of them being the IL-6 antagonist. We were talking about the dose exposure and exposure response. So we do see relationship between exposure response for safety also. The higher expansion has been shown to be correlated with higher grades of CRS. So that's not a surprise. And also we know that it's just not the exposure, but there are several multiple determinants of CRS. So one of them being exposure. I think that I want you to keep that in mind, because we'll again talk about it when we look at this. So we talked about unique aspects of dose exposure, and we do know there are several factors, both intrinsic and extrinsic factors, which will affect this dose exposure response. And this starts from, as I said earlier, from the starting material itself, and we control it so well for other modalities, but that cannot be controlled here because it varies from patient to patient. From the way the T cell will activate, the way that the T cell will respond will vary from one patient to another. Then the conditioning regimen, you need to use conditioning regimen before you give the CAR-Ts, and that also has an impact. And there are several patient factors. So let's just look at them briefly. Let's start from the very basic concept, and we try to identify one of these. The very first thing always you want to look at is the composition of your dosing material. So if that can be controlled, one of the study has shown that if it is controlled for the ratio of CD4 helper cell to the CD8 the cytotoxic cell in a 1-to-1 ratio for CD19 CAR-T, it has been shown that we are able to see much clearer separation of dose exposure here. These are 10-fold different doses, but I showed you earlier example with Kymriah where 10-fold doses were still completely overlapping. But here you're able to see much clearer differentiation. But again, I want to highlight that this was seen in one study, and one size may not fit all, because there are other overwhelming factors which I will talk to you in 1 second, which can diminish this phenomena. So this is not a global phenomena, but if we control the composition, it's possible we can control a better dose exposure. I talked to you about overwhelming factors. So one of the overwhelming factors across CAR-Ts which impact the dose exposure response is tumor burden, a patient's own tumor burden. It's the antigen load, so it's expected. It will more and more engagement, more and more proliferation. And what it shows is clearly with patients at a given same dose at higher tumor burden, you see a differentiation, much higher expansion. And not only that, you see that the expansion is also related with safety concern. In subjects who had much higher tumor burden, you saw that there were much severe CRS and requiring ICU admissions, and that actually is beautifully shown here, that tumor burden is one of the most key important determinant of dose exposure response. Also, I mentioned to you about lymphodepletion therapy. So based on just the choice of regimens, you could also see differences. For example, if you add fludarabine to your regimen for conditioning ahead of infusing the CAR T cells, you see an impact. If fludarabine has been added, there is greater expansion and greater persistence, which has been shown, also translating into efficacy with greater disease-free survival. And so this is one of the another factor which affects dose exposure response. If we look at Kymriah, the very first approved one, if you look at in detail of that, clearly high tumor burden was shown to also result into higher grade of CRS. So this again, this factor is consistent across CAR-Ts. When you have high tumor burden, you will see higher exposure and higher associated CRS grade. They were, when other factors were evaluated, typical ones like body weight. And remember, I talked to you about Kymriah being dosed for based on body weight for subjects who are on a lower end of body weight, but they don't see an effect of body weight on the exposure. And still, this is the dosing is body weight, and we believe this is a little bit where we are being too conservative. And I will show you a little bit again what I mean by that is we are dosing maybe not exactly how the data is showing, but a little bit on conservative side, uh, and trying to be adjusting for a factor which is not really controlling the exposure and the exposure determinant of safety and efficacy, as I showed you. Other extrinsic factors, like I talked to you about controlling the CRS with TOSI treatment, which is the IL-6 antagonist. That one and prior lines of therapies were not shown to be determinant of exposure. But overall, we are seeing again tumor burden as one of the key component of determinant of dose exposure response. And if you look at other manufacturing attributes, I talked to you, there's so many factors could determine. One of the key attributes for manufacturing is the transaction efficiency. And Kymriah has looked at that part, and they were able to show that the transaction efficiency did not impact exposure or expansion. But again, as I say, one size does not fit all. This will need to be characterized across for each development CAR-T because there are so many things which could impact it. But in general, these are some of the things which you want to look at. One unique aspect which I want to quickly touch upon is immunogenicity, which can also impact dose exposure response. Now, immunogenicity has been observed in CAR-T programs, and that can be expected because some of them are murine-based. The receptor is murine-based, and the antibodies are directed against that. However, for all the 6 approved CAR-Ts up to now, the immunogenicity has not been found to be of clinical relevance, and that could be because of the high variability in the dose exposure. And actually, that is highlighted. I showed you some example of labels here where it was shown that it is not clinically relevant. However, the antibodies against the therapy were seen and detected. Now, just not seeing them in the current 6 CAR-Ts may not mean anything because of this reason, which I want to highlight to you, that all of them, the whether anti-CD19 or anti-BCMA, they are B cell targeting. CD19 completely abolishes the B cells. B cells are the one which are the plasma cells which are generating the antibodies. So if your therapies are depleting, again, BCMA also depletes the B cells, the plasma cells, which are generating the antibodies. So just by the design currently, we may have a much less risk, but when we are looking at different antigens which are currently being looked at, and we are looking more into solid tumor space, the immunogenicity could become a big concern. So this is something which we need to pay attention to and be very vigilant about. Now I want to a little bit shift gears. I want to do a time check, and I think I want to talk about the the design and the dose selections for the clinical study for the first human study. So this is what my team helped me, and in the IQ group we looked at all the current ongoing clinical trials in the clinicaltrial.gov for the CAR-Ts, TCR-Ts, allogenic, autologous, and then we tried to summarize and make some understandings out of that vast amount of data. And I want to show you some key highlights of that. If we look at the current first in human dose selection, we see that it is empirical. Pretty much everyone is starting with 1 million cells, and then they're also doing a lot of body weight adjustments here. I've showed you that that may not always be clinically relevant, so everyone is being more conservative, and that could be more from safety perspective. CRS can be a life-threatening safety concern, so I think everyone is being a little bit conservative. They're starting 1 million cells, and then they are body weight adjusting also, or body surface area. Their adjustment is going on, and that may not exactly be the way to always dose them. And so I think a mechanistic understanding of the dose exposure response, as I talked to you, And factors which impact that can be one of the important things to really optimally select these doses. We're not seeing that. Then this is for liquid tumors. If we look at solid tumors, we see autologous, we see TCR-Ts coming into the picture. And if we compare that, we also have allogenic. So if you look at all that, again, we are seeing empirical dose selection. Around 1,000,000 cells are being used across the liquid and solid tumor. And then it doesn't look like it's being guided in a mechanistic manner. If we try to synthesize this data, one thing we were able to see that TCR-Ts are being dosed at a couple of fold higher, around 5-fold, 4 to 5-fold higher than the CAR-T modality. So there is a modality difference between CAR-T and TCR-T, which is all the current vast amount of data is indicating. But if you look at the allogenic versus autologous, we don't see differences in the doses at the starting dose or highest doses. Or if you look across liquid and solid tumor indication, we don't see differences. And that's what really tells us, this is an opportunity where a real mechanistic understanding of dose exposure response should be brought in, of what we expect, what we predict, and really then go with that. And that's something which we'll discuss with you in more detail in subsequent talk when Hardik will come in. Now I want to really talk to you about the optimal design of the clinical study. And this is one of my last slides before I summarize it all. In the clinical design, typically in oncology, if you're starting with very low doses like Mabel, one of the strategy to accelerate that is to start with single dose, single subject cohorts, and that may not really apply for CAR-Ts because I showed you clearly that tumor burden is a very big determinant of dose exposure response for safety also, in addition to just exposure response. And that can be varying from one patient to another. You can have just one subject at starting dose and that can have a low tumor burden, and at the second dose you can have a subject who had a very high tumor burden and you will really underestimate the safety. So that's not the right way to design CAR-T with single subject cohorts. But here is what is more important. Typically in oncology also, our dose escalations are guided by only toxicity, because our key assumption is with the toxicity, the response is also, efficacy is also increasing. But that may not really hold good for CAR-Ts. So if you look at 3+3 design, which no one is currently using, in oncology shouldn't use it, but Bayesian-based designs also are all based on trying to determine the MTD, and that's where the escalations are in trying to determine the 30% DLT rate. But in case of CAR-T, the first dose itself could be efficacious. There is overlap in the dose exposure, and with the increase in doses, you may not really be increasing the efficacy. So this may not be the ideal way to do it, because first dose itself could be fully efficacious. You have seen the approvals are for starting dose to all the way to the top dose that is studying, because they were all efficacious when you start with 1,000,000 cells which are efficacious. So because of that, I think the ideal way to do that is to integrate both safety and efficacy information and not do the typical oncology designs of just using toxicity for dose escalation, combining both together. And one of them is the toxicity and efficacy probability interval method, which integrates both, and that I will suggest. And again, we talked about dose exposure response for understanding both safety and efficacy and factors affecting them, including them understanding that, and then deriving the dose escalation is going to be very important. So with this, I really want to summarize my talk and give you some future directions. If I have to really carry these 5 points I want you to carry back home, is there is a big paradigm shift in the treatment of oncology indications by CAR-Ts, which is brought. And CAR-Ts, because they expand during manufacturing and during the dosing, particularly after dosing, it brings single dosing curative paradigm. But that's not always happening. We have seen that multiple times you need subjects to redose because they lost the expansion and persistence. And in autologous cell therapies, when these are long waiting times, it's not going to be possible. It's extremely difficult in autologous CAR-Ts, which are currently all approved on, to redose. It is a much easier way to do that with allogenic off-the-shelf products. But in autologous cell therapies, when we're looking at the opportunity to improve the CAR-T design, I talked to you about improvements of next generation, the third generation, where you can insert IL-12 transgenes, you can have more optimization of these co-stimulatory domains, which can improve the persistence and expansion. So that's one place to work on in this field for us. The second is the allogenic products need to be accelerated, because I think off-the-shelf products really hold the greatest potential for cellular therapies, but they still are dealing with challenges several types, including from GVHD mechanism to several more, as you can understand, and they need acceleration efforts. So we need to focus on that when we go back and work in our own areas. The third part is we talked about solid tumors are not going to benefit really by the surface antigen recognition. Targeting endogenous antigens using the MHC detection approach, which is the TCR-Ts, I think they are showing great potential, and we need to work harder on bringing them for solid tumor patients who are really not benefiting here. We have a lot of work done in the liquid tumor, but solid tumor is where we need to bring. And this, I think the remaining two bullets are really where clinical pharmacology and pharmacometrics field can really make a difference, where we saw that cell therapies have unique PK/PD properties and multiple factors affect them. Understanding those mechanistically and bringing a mechanistic model-based approach for optimal dose predictions is really going to maximize the patient benefit. And I think that will really help the field, and it's something which we need to implement. We need to then move away from the empirical dose selection, which I showed you, and then just using body weight based adjustments and then just 1 million cells. We need to move away from that. I think we need to bring the model informed drug development, and then typical oncology paradigm of dosing escalations based on toxicity is not going to help CAR-Ts. We to integrate both safety, efficacy, and use model-based approaches to really enhance the patient benefit. And I think that's really my key point. I want to thank you again for your time. And with this, I will give it back to Hardik, because really talk about the model-informed drug development. He's going to take us through what all the modeling approaches really we can use for CAR-Ts. So with this, Hardik, I give it back to you.
Thank you, Vijay. So I will just share my screen. So Vijay, can you see my screen?
Yes.
Okay, perfect. So thank you, Vijay, for going over some of the clinical pharmacology considerations for cell therapies. So before I begin, I just want to thank IQ and the Novel Modalities Working Group for this opportunity to present on their behalf. And this is a perfect segue, right? So what we are going to do over the next few slides is we'll go over some of the PK/PD modeling approaches that are considered for cellular therapies, and will focus on CAR-Ts. Again, to highlight, right, some of these approaches are still exploratory, and they are driven by specific questions. So we'll touch base on some of those aspects. And then finally, towards the end of my section, we'll touch base on some of the translational considerations for cell therapies such as CAR-Ts. So I would like to begin by highlighting a few aspects that were put forth by FDA in a mini review that came out last year. And that mini review was focused on model-informed drug development, MIDD, for novel modalities such as cell and gene therapy. Right. So for those who are not aware, MIDD is the science of developing quantitative models from preclinical and clinical data to help inform decision making in the drug development process. And what this mini review really focused on was not only the opportunities but also the challenges that exist for MIDD for cell and gene therapy. And it was acknowledged, right, that some of these modalities are complex, as was alluded even by Vijay, and also as compared to conventional small and large molecules, we have relatively limited experience with some of these modalities, and that's where also the opportunity is for MIDD to evaluate some of these modalities from a safety and efficacy standpoint, and also considering the hundreds of INDs that have been filed for some of these modalities. There's a lot of scope for building mechanistic models and also to validate some of this parameter and some of the parameter estimates for the mechanistic models. And as Vijay has also highlighted previously, that some of the traditional compartmental models that have been used for small and large molecules are not applicable for cell therapies because of the unique PK. And again, on the schematic on the left, we have just highlighted some of the applications of MIDD in general. for some of these novel modalities, and that can include from dose selection optimization to clinical trial simulation to assess the efficacy, as well as to predict some of the safety aspects in the clinic mechanistically using systems modeling. And in this review, focusing specifically on CAR-Ts, it was also highlighted that it can play a major role in the dose optimization process for CAR-Ts by balancing the safety and efficacy. And one such approach was actually used for some of the regulatory approvals of the CD19 CAR-Ts, including Kymriah, Yescarta, and Tecartus, where a model-based approach was used to understand the safety and efficacy, as well as potential COVID effects of the CRS treating agents such as TOSI. And the model that was used, this is highlighted by the schematic on the right. It's a typical compartmental model. It's not a typical compartmental model. It includes two populations of CAR-Ts as indicated by the effector cells and memory cells. And what this accounts for is until the time until the CAR-T reaches the Tmax, the model is driven by the effector cells, and it has its own expansion. And beyond Tmax, there is a decline in the effector cells, as well as there's a transition from the effector cells to the memory cells. So that's kind of the basic model that was used for some of the evaluations for the previous CAR-Ts. And then this was developed by Stein and from Novartis. And some of us are already aware that this was the first non-linear mixed model to be used for CAR-Ts. Again, one last thing that I would like to highlight, from this mini review was they do emphasize that further research is required into the modeling of the mechanism of action for CAR-Ts, and that can really drive and in terms of improving the dose recommendations for CAR-Ts in specific context, right, with respect to specific patient populations, as well as in predicting this efficacy outcomes. So with that, if you look at the field for the past 3 to 4 years, there have been a lot of models, PKPD models that have been published for this novel modality. And if we want to, summarize them, and most of these models actually are built on a very basic PKPD modeling framework, and that's what is summarized on this slide, right? So there are 3 major components in the modeling framework. The first component is the CAR-T expanders, right? So these are typically cells which have a very fast turnover. They expand when it comes in contact with the target antigen from the tumors, and expand within days, and then they clear out very fast. So that's the first component. And as this cells clear, they also get transitioned into the other cell type, which is the CAR-T persisters, right? So they can persist for a longer period of time, as we have seen with CAR-Ts. They can persist in patients for years. So these are the 2 main components, where the expander cells are short-lived and they represent more of the effector phenotype, whereas the persisters can survive longer and they usually represent more of the memory phenotype. And then the third component over here is the tumor, right? So the tumors within the patients, they can expand and grow in absence of CAR-Ts, but as soon as the expander cells recognize the tumors, not only it aids into the expansion of the expanders, but they also lead to the clearance or the depletion of the tumor cells. And this is really the basic framework that is used in most of the models that have been developed and published so far for CAR-Ts. And depending on the specific questions, we add a certain level of complexity to address that specific question. And then I'll briefly go over what I mean by that in my subsequent slide. So the next two slides we have kind of tried to just give an overview of what are the different models that have been published in the literature, and what are the specific questions, right? We are trying to answer with some of these models with respect to in the discovery and development of CAR-T therapies, right? So we have tried to bucket this in 6 different classes or buckets, right? So the first one is using some of the model-based analysis to characterize the cellular kinetics of CAR-Ts, and here this is basically focusing on the cellular kinetics of the CAR-Ts in the clinic and capturing the multiphasic kinetic profile, as well as evaluating the impact of the extrinsic and the intrinsic factors, as well as the impact of the CRS treating therapies. Also, there have been meta-analysis that have been performed where some of the dose exposure response relationship have been compared across different indications and trials. So that's kind of the one bucket or an application where model-based approaches can help us in characterizing some of the cellular kinetics that we see in the clinic. The second bucket is really the PK/PD modeling of CAR-Ts, right? And this really captures the interaction of CAR-Ts and the tumors. This can be, some of these approaches can be used, preclinically to evaluate the impact of different parameters on the PK/PD, and that can include binding affinity of the CAR-SCFP to the tumor antigen, receptor densities of the target or the antigen, the tumor burden itself, the growth and the killing, the growth of the CAR-Ts, as well as of the tumors, as well as the killing that is mediated by the CAR-Ts. And so some of these parameters can be used in terms of also optimizing the characteristics that we would desire for in the specific CAR-Ts lead selection. And some of these have also been explored to characterize the dose exposure response relationship in the preclinical and the clinical setting. And perhaps the idea also is to use some of these modeling framework for dose predictions, as was also highlighted previously by Vijay. Another bucket that we have included over here is the binding models, right? So again, it's very similar to what we see with CD3 bispecifics, that the binding or the engagement of the CAR-SCFP in this case with the tumor really drives kind of the expansion kinetics of the CAR-Ts as well as the clearance of the tumor. And so some of those binding components, there is a scope of optimizing what kind of an ideal binding affinity we would desire for depending on a specific target, and that is where it can potentially be applied. Some of these modeling tools for binder selection, as well as and in terms of designing the CAR strategy, whether we want AND gating or OR gating, depending on the indication and the antigen distribution. Again, specifically with respect to binding, there are certain nuances and differences between TCR-Ts and CAR, and that Dale will be covering later in this talk as well. And then just continuing, right, the different buckets. The fourth one over here that we have summarized is the biodistribution and trafficking, and this becomes very important, right, in terms of characterizing not only the multiphasic profile of CAR-Ts, but also kind of looking at the biodistribution profiles of some of these therapies in the preclinical and clinical setting. And this becomes more important, especially for solid tumors, where we know like the biodistribution of the therapies can play a role. And also this becomes critical when when the target is also expressed in the normal tissues, and where we expect some of the on-target off-tumor toxicities. So that's kind of the fourth bucket. The fifth bucket, very important, is the CRS, right? As was mentioned previously, that CRS is kind of anticipated with some of these MOAs, and to better understand the cytokine release dynamics, as well as to predict this in patients, can definitely help us in terms of de-risking and also patient stratification. And that's where we can evaluate different parameters, such as dose, tumor burden, exposure, and so on and so forth, in terms of predicting the CRS. And the finally, what we have highlighted on this slide, the sixth bucket is the product-specific characteristics, right? Which was also highlighted previously, right? So that can also have an impact on the overall safety and efficacy in the clinic, and to be able to evaluate the impact of different CD4-CD8 ratios, or even the T cell phenotypic composition on the safety and efficacy. So that's kind of the sixth bucket. So this is sort of the different uh, applications and different questions, which can be addressed with modeling and simulation approaches. Again, the idea over here is not to go into the specifics, but to kind of give you a flavor of what is out there and what's the potential. So with that, in the next couple of slides, I will briefly walk over some case studies very briefly, which we have just picked to kind of highlight some of the applications. So this is one case study which used model-based cellular kinetic analysis of parties in humans, and this was kind of a meta-analysis that was carried out where the cellular kinetic profiles were compared across different trials, and again, individual data sets were leveraged from different clinical trials, including heme and solid tumors. The model structure, again, it's quite similar as what I've described before, and as was also discussed, that it's a multiphasic profile. So each of these phase or the slopes are characterized with some of the rate constants, and essentially, without not going too much details into the modeling part, but emphasizing more on the findings. From this study, right, they did compare the responders and non-responders across different clinical trials, and then they did see that the responders usually had higher Cmax or durable persistence. There's always a question, between hematological malignancies versus solid tumors, and how the cellular kinetics look for this different tumor types. And in this meta-analysis, it was also observed that the Cmax and the proliferation was usually higher for the CAR-Ts for hematological malignancies as compared to solid tumors. And overall, what's also been consistent in the clinical setting from this analysis as well, it was concluded that overall there was a weak correlation for the dose and response. Now, again, the application of some of these approaches is that we can systematically characterize and compare the cellular kinetics across different tumor types and evaluate the different factors that can impact. However, the inherent limitation also is that some of these data is coming from different CAR-Ts, having intended for different targets, different patient population, different manufacturing process, and that all can contribute to the inherent variability. So sometimes it can be very difficult and convoluting to interpret when we do such meta-analysis. So this is another case study, which we have highlighted over here, where systems model was used to characterize the cellular kinetics, but also to characterize the cytokine release in patients. And over here, specifically, the question was, how does the CAR-T doses or the disease burden impact the exposure as well as the cytokines? And the data that was used for this particular case study was from CLL patients with CD19 CAR-T. And again, this is the model structure. It's again based on that basic modeling framework that we described previously, where we have two different populations, effector and memory, and over here they have added additional complexities, right? So over here we have two different compartments, which is peripheral blood as well as the tissue compartment. And essentially how the model works is that there is an interaction of the effector cells with the cells, which is B cells in this case, expressing the target, and that leads to the clearance of the B cells, but then that also leads to the expansion of the effector cells. And there's always and there are rate constants, to describe or characterize the interconversion of the effector to memory, as well as the migration or the migration of the effector or the memory from the two different compartments. And then specifically for this particular model, what was also included was the cytokines So as there is an engagement with the tumors, there is an induction in the production of this cytokines. And the data that was used is also highlighted at the bottom. It's a representative data from one subject. As we can see, the model was able to capture the data, and the data really included was the CAR-T kinetics as well as the cytokine release that were captured in these patients. So overall, the findings from this particular study was that the expansion and efficacy, they were highly correlated to the baseline disease burden and not really the dose. And again, one of the limitation of this particular case study was the limited data availability. So the data that was used to build this model was only from 2 patients, and also some of the more mechanistic aspects were not included. As we can think of for cytokines, there are other cell types as well that are involved, such as the macrophages and so on and so forth, which are not included in this particular case study. However, having said that, some of these can definitely help at a platform level to guide some of the development, as well as the development of parties, as well as personalized dosing, including tumor burden based dosing. So that's kind of where this can be applied. And then finally, the last case study, right? So this is interesting. This was carried out in the preclinical setting, where a PBPK, or a physiologically based PK model, was used to describe the T cell distribution in mice. So essentially, isolating the T cells from the spleen of the mice, and then activating it ex vivo and labeling it, and then injecting again into a melanoma xenograft mice to characterize the distribution. And the model structure is highlighted on the left. It's a typical PBPK model where we have about 12 different compartments resembling the different organs of the body, and then we also had a tumor compartment over here, and they're all connected with the different lymph flows and the blood flows. Within each of these compartments, there is also a transmigration rate of the CAR-Ts going from the vascular space to the extravascular space. And for 3 specific organs, such as the spleen, liver, and kidney, where we anticipate a lot of T cell distribution, retention factors were used to capture the data, as we saw there was some plateau for this organs. So retention factors were used. And then finally, lung was used as a major organ of elimination over here. So this was interesting, and this was interesting because this was kind of the first, one of the first PBPK model that was used to characterize T cell distribution in the mice. And then some of the learnings from this model were actually used later on to develop a PBPK model to characterize the CAR-T distribution, right? And again, without going into the specifics, in contrast to the previous model over here, less number of compartments were used, since due to the availability of the data, as well as one major difference was as is consistent with the literature, the CAR-T elimination was actually estimated from the liver compartment. And again, these are some of the model fits for this model. So we have different compartments, we have different organs over here, and these are the biodistribution profiles for the CAR-Ts. One other important component that I would like to highlight over here was the CAR-T target complexes. So this is kind of the tumor compartment, which was one of the compartments used in the model building process. And within that particular compartment, as I mentioned, there are like 2 subcompartments, vascular and extravascular. So as the CAR-Ts distribute into the tumors, they do transmigrate and move from the vascular to the extravascular space, where they interact with the tumors. And depending on the binding affinity, they interact and form the CAR target complexes that can drive the expansion of the CAR-Ts, but can also drive the killing of the tumors. So that was another assumption that was used over here, was the CAR-T target complexes simultaneously inducing the tumor growth regression, but also the CAR-T cell expansion. And some of these modeling platforms are used to connect to connect the PBPK to the PD aspects in the mouse efficacy study. Again, one of the component I would like to highlight over here is the impact of target expression on the cardiac distribution. I think that is something that needs more investigation, as there's very limited data on some of these aspects, especially in the in vivo setting. Okay, so with that, just to sum up, right? So again, for modeling and simulation for cellular therapies, it's always fit for purpose utilization. And what is the question, and then defining the scope becomes very important, and obviously the data availability becomes important, and depending on the question and the data that is available, there will always be kind of some flexibility in managing certain level of complexities. And then again, some of these approaches can definitely be leveraged, not just in the early discovery space, but also in the late stage preclinical development of CAR-Ts, from characterization of leads to supporting the dose recommendations and beyond. And definitely some of these learnings can be translated to other cell therapy platforms such as allogenic and other cell types as well. So I just have two more slides, before I pass it over to Dale for TCR-T. So I just wanted to touch base on this slides on some of the translational considerations for novel modalities such as CAR-Ts, right? And we have just highlighted a few aspects for us to consider as we develop some of these novel modalities, right? And then these are really specifics to CAR-Ts and more so in the oncology space. So as of now, some of the preclinical studies that are done with cell therapies, especially with CAR-Ts in oncology, the in vitro studies and the in vivo studies that are used are more to establish the proof of concept, right? So they are not really used for any clinical dose selection. Typically, the in vitro assays would include, CAR-T proliferation, tumor cell killing, cytokine release, and so on and so forth. Whereas the in vivo studies would include the xenograft efficacy studies, where we can monitor the expansion and the TGI, or the tumor growth inhibition. And also, variety of models have been explored in the space, and that would range from xenograft, syngeneic to transgenic models. And again, there are advantages and pitfalls with some of these models, which it's beyond the scope of today's presentation. But what I wanted to emphasize is there is really a lack of understanding how to use some of these preclinical data to for human translation. So that is kind of one of the challenge, that how can we leverage some of these preclinical studies to guide the first in human strategy. Again, from a safety standpoint, there are no relevant animal models to evaluate the CAR-T safety, with respect to the on-target or off-target effects. And that's where evaluating the target expression pattern and distribution becomes very important, as well as where there are opportunities, right, where when we are using cross-reactive CAR SCFPs in mouse, then some of the efficacy studies can be leveraged to look at the on-target effects. Again, chip-based protein interaction arrays or heterogeneous-based platforms can be utilized to evaluate some of the non-specific binding and so on and so forth. And also recently, there have been some development in terms of using NHPs and transgenic mouse models to evaluate the safety aspects for CAR-Ts, but they are still in the exploratory stage. And to our understanding, I don't think these have been used to support the filings at this stage. Again, when it comes to cellular therapies, especially CAR-Ts, characterizing the biodistribution in the preclinical and clinical setting becomes very important, especially for indications where the target cells are not in the circulation, right, such as lymphomas or solid tumors, as the cellular kinetics that we observe in the blood may not always be representative of what's happening at the site of action. And especially characterizing biodistribution in some of these studies can also help us to evaluate the potential on-target off-tumor toxicities for a target that is not clean. And finally, as Vijay also highlighted, that we have come a long way in terms of the CAR design, right? So we have first generation, second generation, third generation, and we have newer generations as well. And so it's always a challenge in terms of what should be an ideal CAR design for a specific target or indication, and that's where optimizing becomes a challenge. and some of the considerations are also include the affinity tuning of the CAR SCFE or the binding domain that we use on the CAR for that particular target or the indication. And again, from a product specific aspects, right, how we would want to control the T cell subpopulation, right, with respect to the CD4 CD8 ratios, phenotypic composition, and so on and so forth. So optimizing some of the product specific characteristics becomes important, and that's kind of an opportunity but also a challenge at the same time. So with that, I'll quickly summarize this part of CAR-Ts. As Vijay highlighted, the PK of CAR-Ts is unique and multiphasic because of its living nature, and there have been a lack of consistent dose exposure or dose response relationship, and generally response have been demonstrated to correlate well with the exposure, and from a safety standpoint, the CRS and neurotoxicities they have kind of trended higher at the higher dose levels, but more so with the exposures in some of the clinical studies, and the plethora of patient and product specific factors that can impact the PK/PD aspects of CAR-Ts in the clinic. Again, when it comes to the FIH dose selection for CAR-Ts, allometric scaling is not relevant. Currently, the strategy that has been employed is really using the previous clinical experience with the related products, but again, it's very highly empirical approach, and it's not ideal since there could be a lot of differences between different patients, as well as there would be target specific considerations, and that's where mechanistic PK/PD modeling can definitely help in terms of those optimization. However, having said that, we do need a lot of preclinical to clinical and reverse translation efforts in this space to validate some of the mechanistic modeling approaches for dose selection. And as we move towards the next generation of cell therapies, as was also highlighted previously, we have a lot of success with some of the heme indications, but there's a lot of scope for improvement in the solid tumor space, and that's where some of the other cell type therapy, such as the CAR-NK or TCR-T, are also being explored and can help to navigate that space. And with that, I think it's a perfect segue to pass this to Dale, who will actually talk about some of the clinical pharmacology considerations for TCR-T therapies. So let me pass over this to Dale.
All right, hi everybody. So my name is Dale Miles, and so right now we'll shift gears a little bit and talk about TCR-T therapies. Now, a lot of the really great work that was just described by Vijay and Hardik will translate very nicely to TCR-T. And my goal is not to recapitulate that, but rather just to talk about the unique challenges and opportunities for TCR-T, both in terms of the therapy itself, as well as some unique clinical pharmacology strategies to optimize these TCR-T therapies. So let's jump right into it. So we have TCR-T over here on the left, CAR-T over here on the right. And one thing you can see immediately different is the TCR is recognizing the peptide, which is this sort of light purple. It's usually 9 to 10 amino acids in the context of MHC, are also sometimes called HLA. And that allows this TCR to access virtually any intracellular protein that's processed and presented via this mechanism. So a much richer repertoire of potential targets via the TCR versus the CAR-T. But that does come with a limitation, and that is that these presentations are HLA restricted. It's only recognized in the context of a particular peptide MHC, and we'll have a lot more to say about that in a minute. Now, if we jump over here and look at CAR-T, you'll see if you take the variable region of the heavy and light chain of an antibody, join it with this sort of flexible linker, amazingly, it binds antigen similar as if it were an antibody. However, you'll also notice that it's limited, at least largely limited, to surface antigens. So if the antigen is not presented on the cell surface, this construct can't recognize it. That said, there is a lot of work to try and target these toward a peptide MHC for obvious reasons, and some good preliminary proof of concept work has been done. And I have a reference below about that. The other advantage here for CAR-T is there's no HLA restriction. Then let's look at co-stimulation. That's another difference. So you can see here over for the TCR-T, CD28, for example, co-stimulation is using endogenous co-stimulation, which is already built into the cell. Whereas for a CAR-T, we actually hardwire or engineer in the CD28 co-stimulation. And because of that, potentially, you might expect more activation in terms of tumor cell killing and T cell activity and possibly CRS compared to TCR-T, which uses these endogenous mechanisms, although I think that still needs to be proven, but theoretically that could be a difference. And then let's look at the binding as another interesting difference. So you can see here over here for TCR-T lower affinity in the micromolar range, whereas much higher affinity, antibody-like affinity for the CAR-T in the nanomolar range. But then on the flip side, the TCR can recognize is very sensitive, just 4 or more peptide MHC complexes is enough to trigger cell killing, whereas over here on the CAR-T side, you need at least an order of magnitude higher antigen density in general. And then for both therapies, even higher antigen density to trigger cytokine release. And so why is this sort of disparity between affinity and sensitivity? And that could be that avidity is important, and avidity, and we'll have a lot more to say about that, is the strength of multiple peptide MHC TCR interactions and the contribution of the combination of those to the overall effect. So the other big difference, obviously, 6 approved therapies for the CAR-T and no approved therapies yet for the TCR-T, but I think a lot of excitement coming. And I might highlight one more difference, and that is that because of this MHC HLA restriction, you can have a CD8 TCR targeting MHC1 or CD4 TCR targeting MHC2, but you can't really do with the same TCR target using both cells at the same time. Okay, TCR-T and CAR-T have very similar manufacturing steps, just like what you saw before. The only difference would be here in the genetic engineering piece. So we're putting in a TCR, potentially knocking out the endogenous TCR rather than putting in the CAR construct at this step. But similar to other, it could be allogenic or autologous. Also, we could differentiate stem cells to achieve a desired cell type. So here are top targets. So you can see, and pay attention to the x-axis. So this is 250 and 60 over here. So the scales are different. But you can see that CAR-T, which is this light blue, clearly dominating in heme. Not surprisingly, the approved target, CD19 and BCMA, leading the pack. TCR-T, which is in white, it actually is being studied in heme, but you almost need a magnifying glass to see it. But those studies are ongoing. But not surprisingly, in solid tumors where TCR-T might bring some clear advantage, you can see that there is a better presence of TCR-T, with the NY-ESO cancer testis antigen sort of being the most prominent. So tumor antigens, I'll talk about those for a minute. They can be shared or they can be private. So shared means across individuals, and even across individuals, these could be subcategorized to tumor-associated antigens, which are elevated in the tumor. So the cancer testis antigen that I just showed previously, EGFR, they're not exclusively in the tumor, but they're higher in the tumor. There can also be tumor-specific antigens that are shared across individuals. So these are indeed unique to the tumor and also shared across individuals. And then on the other side are private antigens within an individual. And the best example of these would be the tumor-specific antigens that are unique to the tumor. For example, a somatic mutation that makes a cell cancerous could lead to an antigen that one could then detect and be very specific to the tumor. And TCR-T therapy would be very good for picking that out, whereas CAR-T might have challenges recognizing such an antigen. So let's go through some of the unique challenges for TCR-T. Mispairing is certainly one of them. So you can see over here an engineered TCR with an alpha and a beta component, endogenous TCR, also an alpha and beta component, and these have been just striped out to show the difference with hash lines. But as the cell assembles a TCR at the surface, it could potentially associate this alpha with that beta or vice versa, and get these potentially mispaired TCRs in addition to the ones that you are intending to create. So this is undesirable, obviously, because this might have different or unknown affinity from what you were originally intending, and perhaps an unexpected toxicity. So we generally don't want this to happen. There's some engineering ways to get around this, but one simple way, a simpler way, is just to knock out the endogenous TCR. For example, using a CRISPR-Cas9 system could be done to do that. The other thing I'll mention is that these endogenous TCRs also compete for assembly with the CD3 units. So by knocking them out, we would no longer have that competition. And then this is the big, I guess, challenge for TCR-T therapy is this HLA restriction. Tumor-related peptides or antigens must bind HLA to be presented at the cell surface. And the peptide affinity for the different HLA haplotypes, and there are many different haplotypes, can vary widely. And the HLA haplotype of all these different types can vary across geographic regions. So what that means is you have to consider both how well your TCR binds the peptide and also the haplotype frequency in the population that you intend to treat in order to come up with what you think is an optimal therapy. And even in the best case, if you take the HLA-A0201, which is the most frequent HLA haplotype, that's still found in typically less than 50% of cancer patients. So if you have a single TCR therapy that you're taking forward, it's TCR target, you're already limited to half the population from the get-go. And this HLA and antigen have to be considered both for clinical as well as your non-clinical studies. And obviously, human HLA does not translate over to mouse HLA. So quickly, native versus enhanced TCRs. So potentially, you can improve anti-tumor activity by enhancing. And why do we do that? So the native TCR affinity for some tumor antigens, and you could think of a tumor antigen being a little bit more like self compared to, for example, a viral protein. So sort of inherently low native TCR affinity, at least for some of these antigens. And the thought was by making some simple amino acid substitutions, tweaking or fine-tuning or increasing that affinity for the antigen. So that's been done. It's also been shown if the binding is too strong, that can actually be counterproductive. So more is not always better. And then also when you do that, you have to consider that it could potentially impact safety and immunogenicity. So you're now changing from an endogenous TCR to something a little bit different that your body might recognize differently. There's a number of different modeling approaches to TCR-T therapies. Those are covered very nicely by Hardik. We can go to the more empirical models like the Stein model. So it separates into effector and memory subsets based on the cellular kinetic profile, but without actually directly measuring those. And this model can actually be quite useful. So if you want to compare or interrogate product characteristics for their impact on cellular kinetics, you could use this type of a model. Or even if you wanted to take cellular kinetics and relate to, say, efficacy, you could do that as well. So if you have the clinical data with very few assumptions, this model can be quite powerful to draw some important conclusions and insights. On the other hand, if you need more mechanistic understanding, for example, combination with another immunotherapy or biodistribution to places other than the peripheral blood, if those components are important, then some of these more mechanistic models can be helpful. And here you can see there's a broad spectrum of those, ranging from semi-mechanistic all the way to even fully mechanistic. Just realize that the more mechanistic the model, the more bottom-up data that you're going to need, and that might often be missing for a CAR-T therapy. So this dual modeling approach is one that we have and others have taken, where depending on the question, you might use more empirical, or if you're looking at combinations with complex interdependent pathways, we might use something a little more mechanistic. The critical mechanisms, I think, have all been talked about: expansion, trafficking, the antigen-triggered activation, apoptosis, biodistribution. The one thing I'll note here is if you do knock out the endogenous TCR, like I showed earlier, and then you fail to knock in your neo or transgenic TCR, you can have what are double negative cells. So these are T cells that have no TCR at all. So there might be some regulatory as well as company interest in understanding a little bit how those behave and if they have any negative impact. So those could be monitored in addition to the typical phenotypic subsets like stem cell memory and central memory. Unique aspects for modeling TCR-T is first of all, functional avidity and cell killing instead of a binding affinity. I have a whole slide on that in just a minute. Endogenous co-stimulatory domains rather than the hard-coded co-stimulatory for CAR-T. Potentially different biodistribution requirements, especially if you're targeting solid tumors, and then high reliance on clinical and in vitro data. Non-clinical data, even harder to translate than CAR-T, and I'll show you why that is in a minute. So affinity and avidity are more challenging for TCR-T versus CAR-T. So affinity is just the strength of a single TCR-P-MHC interaction, and we know for TCR-T that the binding half-life K-off is important. Additionally, avidity is the strength of multiple engagements, including the effect of co-receptors. Now, this can and ultimately probably will ultimately all be modeled and figured out, but right now I would say we're still finding our way there a little bit. So sometimes we sort of jump right to the end and look at functional avidity, and that is as we vary concentrations of our peptide antigen over a range, we just look at the EC50 for a T cell activation or T cell killing, and then try and translate that, rather than work out some of the details here, which I think still needs a little bit more science, a little bit more work to get to understand well. Unlike CAR-T, it's challenging to measure and interpret TCR-T binding to the target. So there's some divergence between affinity and T cell affinity activity, just like I described above, affinity and activity not as well correlated as for CAR-T. And so dextramers and hexamers are often used to try and incorporate that avidity component. So you could see, for example, here, here's a tetramer. So you have a construct that has multiple peptide MHC that can then can bind multiple sites on a TCR cell at the same time. And in fact, if you're doing flow cytometry, you actually need this to get tight enough binding to really get a good flow signal. So that avidity component is important. And then the other thing is this peptide, which you can probably barely see, this little red dot is the peptide on the blue MHC from the cancer cell. It's actually very difficult to measure that surface expression of peptide MHC. It's not impossible, but it's very hard to do. And in many cases, if you don't have those data, you're shooting in the dark a little bit because you're trying to design a TCR therapy for a surface expressed target that you really can't see or measure at all. So in vitro TCR-T are difficult to translate. Syngeneic could potentially elucidate off-target toxicities, but does not allow evaluation of the human TCR-T cell product for PMHC, HLA considerations, and nor does it recapitulate the human immune system. Xenograft actually can work well for proof of concept efficacy, but it's hard to delineate between rejection, allogenic response, and actual TCR-T efficacy. So again, very hard to extrapolate or translate to dosing recommendations beyond just proof of concept efficacy. Transgenic and humanized mice are being developed. This overcomes some of those limitations, but I think those still need a little bit more work before they're ready for prime time. So overall, a high incidence of graft versus host disease, TCR-T rejection, lack of matching PMHC is a major concern for many of these non-clinical models, and current in vivo models support proof of concept, but not clinical dose selection. So this means we have an opportunity to fully leverage, and not just an opportunity, but a need to really fully leverage our clinical data. You can see here some cell kinetics data was combined across multiple indications in a pooled analysis to try and understand what was driving response and responders and non-responders, and also some modeling approaches, as Hardik described, to take relatively limited clinical data and then look at T cell phenotypes and other factors of the infusion product to try and better understand its role in driving safety and efficacy. So the bottom line, even more so than CAR-T, we have a high reliance on in vitro data for supporting our first in human studies. This could include in vitro pharmacology data, safety assessment. I don't have time to go through these today because of limitations of time, but these are interesting read if you want to look at these on your own time, of how we can de-risk to really a large degree, at least from a safety perspective, these TCR-T therapies based on in vitro assays alone. And then based on a review of published TCR-T therapies for solid tumors, typically we see activity across a range of doses, 1 times 10 to the 9th up to 1.23 times 10 to the 11th. Many of these clinical trials a dose on total T cells, which includes the TCR-T cells, but non-engineered cells as well. And sometimes that's just due to manufacturing feasibility considerations. TCR-T doses are typically higher than CAR-T doses, as Vijay mentioned. And there's occasional but not consistent dose exposure relationships, and that's often due to small n. And there's probably a lot more that we could do and learn around those relationships as we get more data available. So to conclude, TCR-T therapy is promising, expanding new modality. Importantly, it has a potential to target a wider repertoire of intracellular proteins and enable personalized therapies, but it's limited by HLA restrictions. Non-clinical data are very difficult to translate, high reliance on in vitro screens, and we need to fully leverage clinical data as we continue to develop our abilities in the non-clinical translation space. And a questions-based approach to modeling drives the appropriate level of model complexity. So with that, I am going to conclude and turn my presentation back over to Hardik. I'm sorry, to Nag.
Thanks a lot, Dale. Thanks a lot, Vijay, Hardik, and Dale. Thanks a lot for the fantastic talks. I would like to get the question and answer session rolling. There are a couple of questions which came up, and I would like to pose them to all the three of you. What are some things to consider when interpreting dose exposure and dose response relationship, be it for CAR-Ts or TCR-Ts? I mean, clearly there aren't a lot of good preclinical models, etc. So how do you go about that? I mean, Vijay, you want to take it first, and then, Dale and Hardik can join in about TCR-T and etc. Please go ahead.
Absolutely, Nag. And first of all, we want to say thanks to everyone for staying through the talk, and we want to answer the question. I think one of the important things for the dose exposure and response understanding is to really realize that there is more than what typically you see for small molecule and large molecule as a source of variability in it. And you're going to see those overlaps in the exposure, because as I said, right, it's just not the patient factors, which typically are the focus for all therapies, small molecule, large molecules. But here we have to look at manufacturing, the raw materials which are used, which are very variable. So all those factors add to the variability, and is going to be, so not to be surprised when you see a lack of dose exposure. But as all three of us highlighted, right, there is a clear relationship between exposure and response. And I think that understanding and trying to understand the factors which cause this dose exposure differences can really allow us to predict, like the models what Hardik talked about, to really design the dosing and move away from that empirical dosing, which everyone is currently doing, and to move towards a mechanistic model informed dosing. I think those are the key concept which we need to take forward. And I'll invite Dale and Hardik to add to this answer.
Go ahead.
That's really great, Vijay. Certainly agree with all of that. And I would say just from a very simplistic point of view, when you read the literature and you see a dose response, one of the first things that one should verify is were these doses assigned retrospectively or prospectively. And what I mean by that is, for some patients, we may not be able to manufacture the intended target dose. And if those patients are then assigned to a lower dose level, then that could potentially bias any dose exposure relationship, because we know that patients' cells who don't expand very well in vitro often don't expand very well in vivo as well. So the first caution would be, how do we treat patients that for which we're not able to manufacture the intended target dose? If they are put in a lower dose cohort, maybe they should be separated out for analysis so that they don't confound interpretation. And then the second thing, as Vijay described, I think is very important, is understanding the key drivers. So if dose is not the key driver of even cellular kinetics, then spending a lot of time on evaluating the dose is probably not going to get us very far. We need to find out what's driving the cellular kinetics. And is it some component of the dose? Is it a certain phenotype of T cells? Like what's driving that exposure relationship? And then I think we have, and hopefully those are things that we can control as a clinical pharmacologist. And if it's a key driver that we can control, then we have ability to optimize the therapy.
So what are some of the reasons for that? I mean, you're talking about the lack of the relationship, lack of dose exposure response relationship, and what are some of the reasons for that, if you don't mind going into it?
Yeah, I think we talked. Yeah. Go ahead, Dale. Go ahead.
I was just going to say it's because you put the same number of cells in 10 different patients, and they can expand very differently by a factor of tenfold. So it's not what you put in the patient, it's what they expand to after they go into the patient that determines your exposure. And so dose is just one of many components that determines that expansion. Vijay highlighted some of those things: tumor burden, the fitness of the cells that you put in. There are many different factors that determine that expansion, and I think that's why the dose exposure relationship looks all over the board, is because we have to account for those other factors. But Vijay, did you want to add to that?
No, I think that's perfectly as I was going to say. Thank you so much, Dale. Other thing, just you asked this key question is why dose exposure is not seen and we are able to characterize well for other programs and not here is that additional factor which we all three have talked about, that you have patient factor, manufacturing factor, starting material. Now think about like therapeutic proteins or small molecules, right? How well we control the starting materials. They go to the gold standard. Now here, patient to patient, you cannot control it. T cell will expand differently. T cells will behave differently from a patient to patient. So all those factors come in. So I think everyone has to keep this thing in key mind, that the variability will be humongous and you will have to understand. So lack of dose exposure relationship need to be looked very carefully, and you need to determine the predictors of that, which we are going to affect because exposure response is very strong. So you need to understand, be able to predict the factors which are going to impact it. That really is the key where the field will need to move on. And I think it is all three of us has said the same thing. And I realize I have not given chance to Hardik to say anything to it. Hardik, you want to add something to what we have been saying?
No, I think very well summarized here, and Vijay. I don't think I have anything to add. Just a minor point also, like we are also dosing in the same range, right? Like, so there's not a wide range that we do with some of these therapies. So something to keep in mind, right? Like when we are evaluating some of these aspects, that's all pretty much in the same range.
A very naive question coming from me, maybe to extend the. I'm just thinking, are there any behavioral, nutritional, food-related, factors which can impact such expansion? I mean, when I say behavioral, what I'm talking about is stress, job-related stress, etc., etc. Are there any co-medications which can impact such expansion of the cells post-administration? Food-related, anything? I mean, I'm just curious what is going through, some of your minds who are involved in this. And I mean, if the audience has any thoughts, please put it in the chat. I would love to hear from them. We all would love to hear from you. Any of the three of you, would you like to speak to that? I'm just thinking
I think it's a very, very good question, because I think this is the clinical relevance of this, right? So in general, typical factors like food, which will commonly affect the small molecules which are orally administered, are not known to be, in this case, not expected to be. One of the common co-medication which is given, as I mentioned earlier, is the tocilizumab, the IL-6 antagonist, which is given, and that's one of the key factors to determine if it's going to impact the dose exposure response, right? Because it's IL-6, when you're blocking IL-6, you could expect that the proliferation could be expected. So when it showed some data from one of the CAR-Ts where it was not found to be a cofactor, but again, I like to say this, and it's very important, which we want to take as a carry message home, is one size will not fit all. So I think evaluating during the development of each cell therapy, the factors which will determine, learning from prior experience, like we have tried to summarize and distill this knowledge currently up to available. But again, every molecule should be evaluated, and one size will not fit all. I will say that, and I will invite my other co-speakers to talk on it.
I have a question from Mohammed in the audience, Mohammed Hussein in the audience. I'll pose it to the three of you. In FIH, how are expansion and persistence taken into account for dose selection and dosing regimen?
So currently not, right? I showed you the whole clinical trial .gov picture readout and each of the modality, each of the therapies, and autologous and allogenic, TCR-T CAR-T. TCR-T CAR-T, expectedly there were differences in dose, but nothing else. So what you are seeing is people are starting empirically, starting with 1 million, and they are actually escalating the doses. Typically, if there are not a huge safety concerns based on prior experience with that target, for example, a previous bispecific T-cell engager hitting that target, then typically half log scale 3-fold escalation happening, and everyone is doing like those simply. But integrating the dose exposure response, I think that's the next level of field which we talked about that we should be doing. That's how the guiding of the dosing which will really happen. So it's very different paradigm than, we don't want PK based escalation unless there's a safety concern in typical therapeutics. But in cellular therapies, I think this may be a place where you want to include PK exposure and start determining those factors based on prior knowledge. So currently not. Currently, I think, as I said, they're being done a typical way, but I think the field need to move away from it.
Right, and that is just to add, right? I mean, that is where even for FIH, right, the preclinical data is a lot of question mark, right? How do we utilize some of those data for the dose selection? So, I think right now the approach is really to compare, like a similar target or a similar product that is used in a similar patient population. I think that is the best that is done, and then to leverage some of those clinical precedents, right, from a related products. But again, if you look at the recent guidelines that came out last month, FDA in the draft guidelines, I do emphasize that like the sponsors should be careful while leveraging any other CAR-T product, because there could be target specific differences as well. So yeah, so I mean, just to summarize, it's very empirical at this stage, and that is where perhaps we can bring into some of the modeling aspects as well to guide the FIH dosing.
We have two more questions. The first question is from Dilki Vikrama Sorry if I've mispronounced your name. It follows along what we were discussing. Maybe any food supplements that improve immune cell health in general may help in improving the response. Just a thought. I mean, that's a thought. Maybe we can respond to it. We don't need to respond to it. Here's one question from me. Would there be any ethnic aspects that you need to think about when these cells replicate or expand in vivo following administration? Would there be any genetic, when I say ethnic, I mean genetic aspects that you need to think of, really? If you can comment.
It's a very important question, and I'm glad they asked, right? So I think, as we say, right, these therapies are live biologic. So any factors could impact it more than what typically is seen for small molecule, large molecule. So I think these need to be evaluated. I believe that, if you are able to enhance the immune system, it should help just based on, because these are immune therapies and live biologic. I have not seen the data specifically, but that could help. The other part, what was the second part of the question?
I mean, the first one was a thought. It was just that, maybe any food or supplements that improve the immune cell health may help in improving the response.
So it is possible, yeah. And the second was about ethnic differences, right? It was asked, and I think that's a very important clin pharm question, which we are always spend a lot of time looking at it. It is possible, it could be, right? It's again same way like when very early small molecule or large molecule came in, we thought that one size will fit all, but now we understand the differences better. And I think that's how the field will go for cell therapies also. There could be potentially, if there are any ethnic differences which could impact immune system and the way expansion happens, it could potentially be, but that at this stage it's speculative for me. But I will ask, anything Hardik and Dale would want to add.
Yeah, I don't have much to add other than I do believe race has been evaluated in the approved therapies so far, and I don't know how broadly sampled race really was sampled in those studies, so I can't really comment, but I don't recall race coming out. Age sometimes will come out. So obviously, an older patient may have less fit T cells, so you'll sometimes see age come out as a sort of an intrinsic factor that impacts sort of the overall therapy. I don't recall seeing race, although I may have missed it, and I don't know how well the studies actually interrogated race.
There's a question from Edith Tarsha from AbbVie. Is TCR-T toxicity profile different from CAR-T?
So there's two types of toxicities, right? So CRS, and I don't think that any of this is proven because it's hard to do head-to-head comparisons between the two therapies. But I think the thought is, and the data suggests, that CRS is probably less for a TCR-T compared to a CAR-T, and that could be for many reasons. So CAR-Ts right now, are at least the CD19, are targeting circulating disease. They're seeing a lot of target. They also have this sort of hardwired co-stimulation, and for one or both of those reasons, we see a lot of CRS with the CAR-Ts. TCR-Ts, we can still see CRS, but it seems to be somewhat attenuated. But again, I don't know that we have the right head-to-head study to really say that with any certainty. And then the other toxicity is just your on-target off-tumor toxicity. And of course, that will be different, not just between CAR-T, but that could be different from one TCR-T to another. So, and it depends on your antigen. So if you have an antigen that's truly unique to the tumor, then that shouldn't be an issue. But oftentimes we don't. If we're looking at a cancer testis antigen or some other antigen that's also expressed to some degree on normal tissue, then for that particular therapy, you may see sometimes serious toxicity, and there's examples of this in the literature where patients actually died because of an on-target off-tumor toxicity that wasn't necessarily captured before it went into patients. And so that's why that in vitro screening is so important. So you can capture a lot of those with the right in vitro screens to minimize. You can't reduce it down to zero, but you can minimize those kinds of toxicities with good in vitro screens. So the bottom line is there going to be different toxicities from that perspective? CRS probably will be less, but I think that remains to be proven definitively.
Thank you, Dale. There's another question from Suresh Swaminathan. Basically, it's about, can you comment on the proliferation capacity of a CAR T cell post-activation? You know, like how many cell cycles can a single CAR T undergo? Is it like, can we estimate it? Is it finite, or, you know, until it is cleared from the systemic circulation, along those lines? Vijay, Hardik?
Yeah, I think this is very variable, as we said, right? One size doesn't fit all, but you know, up to 10 years persistence had been talked about and it has been detected. So for CAR Ts, so I think we believe that, you know, CAR T therapy based on, and then again, it will depend on the patient, it will depend on indication, it will depend upon the way the CAR T is designed. But there could be longer persistence, which may mean that, you know, this therapy being a live biologic could exist and could continue proliferating on its own, and that's the power of it. So yes, it will exactly quantifying, you know, how much it will depend, will all, we talked about so many factors which determine it. So there is no quantifying number which can be put, at least in my thinking, but yes, they can persist. But at the same time, you saw where there's failure to launch in patients, you know, the half-life will be like 4 days, 5 days, and then way different than patients who respond. So response versus non-response will be very different, but then how many times it will persist, that will be very variable. That's what will be my 2 cents. And I just want to make sure that time check, we have gone very far, but I want Dale to finish his thoughts. Are we okay to carry forward now, because I think we went 15 minutes over, but I want to have Dale, I don't want to miss what Dale wanted to add.
We still have
just really, sorry, go ahead, Doug.
No, we still have quite a few attendees on board, so please go ahead, finish it.
All right, I was just going to mention that the phenotypic composition of the cells changes after you infuse the cells. So you have some cells you differentiate, expand very rapidly, but then those cells die off. Those are sort of your effector memory cells or effector cells. And then you'll have other phenotypes like your stem cell memory and central memory cells. Those cells might expand less quickly, but they're going to persist and continue to expand at a slower rate over a long period of time. And so it's not a simple answer to how many divisions is the cell going to undergo. It's going to depend on the phenotype and how it differentiates, which is a complex process. And then the other thing to point is we do see a lot of expansion. So I think it's 10 or even sometimes 100-fold more cells from what you put in to what you get at Cmax, for example. So there's a lot of action, at least early on. A lot of those cells will die off, they'll apoptose, contract, and then there'll be a certain subset that will persist for a very long time.
Thank you.
No, so those are really great points. Just one final thing to add over there, right? But still, I mean, human body is still a finite system, right? So I mean, it's not that it will be even at the max, right? Like there will be some finite capacity, and also you don't have to, we also are doing lipid depletion, so that creates extra space as well for to accompany maximum. But then again, like the top, there would be like some capping, right? Because our system as such is finite. But as Dale alluded, we have seen like hundreds or even thousands of fold higher numbers as compared to what we dose at the Cmax. So definitely we see a lot of expansion, and then there are the product and patient specific characteristics that drive the final fate.
Fantastic. Thank you. I have
We're
one
really
last.
glad everyone is staying and giving us extra time, because I think we have gone way over time. So thank you for allowing us that. We're very happy to stay. I'm very happy to stay and continue addressing the answers.
Thank you. One last question. When you're conducting, putting together your trial design, etc., do you take into account if any of the co-medications or comorbidities can alter the expression of the co-stimulatory domains that you have, or what they might end up binding to within the patient, etc., if there's upregulation or a downregulation? Do you take that into account in your clinical trial design? How do you even do that?
I can share my perspective and then I'll ask the speakers to add. So currently, we follow very strict inclusion-exclusion criteria in our clinical trials, and I think everyone does that because the clinical trials have to be governed by that. But one thing I want to be very clear about that, right, is one aspect which really determines the safety profile and the exposure. All of that is the patient-to-patient variability in the tumor burden in a big manner. So I think having we don't control for the factor which we already know is a very big determinant of those exposure response. We allow everyone who qualifies based on inclusion exclusion criteria currently, because it's empirical. We are going to collect the data and see it. Having said that, we do implement design aspect about it, right? For example, we do not support single subject dosings because it can be very misguiding on the safety. So those type of factors which we already are known are being incorporated in the design like this, but not we are not controlling for one patient over another, and that's what my experience will be. But I will ask other speakers to add also.
Hardik, Dale?
Yeah, really nothing to add. I think the big drivers of variability, or as Vijay mentioned, there could potentially be some of these other interactions, but if it's a drug that's not directly impacting your immune system, I would be probably less concerned about that over some of these more factors that we know are big drivers of variability.
Also, some of these patients are, late stage patients, right? So, it's always that balance of safety versus efficacy, and what this will add to the patient's lives. So,
Can do. Thank you. I do not have any other questions, so I would like to thank the three speakers, IQ, as well as all the audience for patiently being here and providing three speakers an opportunity to present on these topics which are pretty close and dear to their heart today. Thanks again, everyone. And with that, I will close out the webinar.
Thank you, Nag. And we want to say thanks to everyone for staying over time and giving us the extra time. We definitely took way more than we should have, but we don't want to end it without saying thank you for your patience and staying with us. And thank you to my other co-speakers. I really appreciate all that work.
Thanks again, everyone.
Thank you, everyone.
Bye-bye.